Hello friends, today we will see few questions from biological classification. First question: artificial system of classification was first used by Linnaeus, Bentham and Hooker, Pliny the Elder, Aristotle. The correct answer is Linnaeus. Carolus Linnaeus is the one who gave this artificial system of classification based on the observable characters based on the observable characters. So he just observed the number, size, and the union of carpel and stamens, and based on this, he classified the plants totally into 24 classes. So the correct answer is Linnaeus. Second question: which scientist proposed the three kingdom classification? Copeland, Bentham and Hooker, Ernst Haeckel, R.H. Whittaker. The correct answer is Ernst Haeckel. So Ernst Haeckel is the one who proposed the three kingdom classification in the year 1866. So the three kingdoms are, first one is Protista. So in Protista, he has included all the unicellular organisms into Protista. And the second one is Plantae, all the plants, and the third one is the Animalia. So these are the three kingdom classifications. Third question: which of the following is not a criteria used by R.H. Whittaker to classify the organisms? Complexity of cell structure, complexity of body structure, mode of nutrition, nature of habitat. The correct answer is option number D, nature of habitat. This nature of habitat is not included as a criteria. It is not included as a criteria in the R.H. Whittaker's classification. Along with these three, there are two more, that is reproduction and as well as phylogenetic relationship. Phylogenetic relationship with the other organisms is also a criteria that is included in the R.H. Whittaker's classification. So the cell structure, body structure, mode of nutrition, reproduction and phylogenetic relationship are the 5 criteria that are used for R.H. Whittaker's classification. Fourth question: three domain system is given by Copeland, Bentham and Hooker, Carl Woese, Aristotle. The correct answer is Carl Woese. So he has given this three domain system in the year 1977. This is based on the sequence of rRNA genes. So based on this rRNA genes, he has divided or classified the organisms into three domain systems. What are the three domain system? That is archaea, then bacteria, and the last one is eukarya. So these archaea and bacteria are the prokaryotes, and eukarya has eukaryotes. Fifth question: the book Genera Plantarum was written by Bessie, Bentham and Hooker, Engler, Hutchinson. The correct answer is option B, Bentham and Hooker. They have written this Genera Plantarum. In this book, they have mainly focused on the seed bearing plants. Those are the phanerogams. So these are the seed bearing plants. They have classified this phanerogams into three classes. So that is dicotyledons, monocotyledons, and then the gymnosperms. So these are the three classes they have divided into. This is basically again based on morphological characters. Morphological characters only, observable characters. So that's why we call this as a natural system of classification. So if anyone asks who gave the natural system of classification, it will be Bentham and Hooker. Next question. A system of classification in which a large number of traits are considered is phylogenetic system, artificial system, synthetic system, natural system. The correct answer is option D, natural system. Natural system of classification is done based on morphology, anatomy, cytology, embryology, and phylogeny is also considered. So this is the phylogeny means evolutionary relationships. So who gave this natural system? That is Bentham and Hooker in their book called Genera Plantarum. Seventh question. The phylogenetic system of classification was put forth by Carolus Linnaeus, Aristotle, Copeland, Adolf Engler and Karl Brandt. The correct answer is Adolf Engler and Karl Brandt. They both are German botanists, they both belong to German, and they have given this phylogenetic system of classification based on evolutionary and genetic relationships. So based on this they have put forth this phylogenetic classification. This is also called as cladistic classification. Cladistic classification or phylogenetic system of classification, both are the same. Eighth question. In Whittaker's 5 kingdom classification, eukaryotes are assigned to only 4 of 5 kingdoms, only 3 of 5 kingdoms, only 2 of 5 kingdoms, all of the 5 kingdoms. First, let's see what are these 5 kingdoms. We have Monera, the first one, then Protista, then fungi, plantae, and the last one is the Animalia. So these are the 5 kingdoms. So we know very well that the Monera, the first one is prokaryotes, right? The first one includes prokaryotes and remaining all the 4 include the eukaryotes. So now we can answer the 5 kingdom classification. The eukaryotes are assigned to only 4 of the 5 kingdoms. That is option number A. Only 4 of the 5 kingdoms. The one kingdom which is not assigned into eukaryotes is the Monera. Ninth question. Which one belongs to Monera? Amoeba, Escherichia, Gelidium, Spirogyra. The correct answer is Escherichia, right? Monera, it solely has only bacteria. It has only the bacteria. So if you see, amoeba comes under which group? Amoeba comes under protozoan. Protozoan which comes under Protista again. Then Gelidium comes under the algae which comes under plantae. And then Spirogyra also comes under algae. This is green algae and that is the red algae, right, which comes under plantae. So the correct answer, only the Escherichia is the bacteria which comes under the Monera. So the correct answer is, it's the option number B, Escherichia. The question. Absorptive heterotrophic nutrition is exhibited by algae, fungi, bryophytes, bacteria. First, let's see what is this absorptive heterotrophic nutrition. So here in this, the organism will secrete the digestive enzymes and convert the complex organic matter into the simple matter, and then this will be absorbed into the body. So this is called as absorptive heterotrophic nutrition. This you can see in the fungi. So the correct answer is option B, fungi. 11th question. X is a spirally coiled filamentous blue-green algae covered by a sheath of mucilage. Eggs can be easily cultivated in tanks and can be used as a palatable protein-rich food supplement for humans and animals. Identify eggs. Nostoc, Gunnera, Trifolium, Spirulina. The correct answer is Spirulina. Spirulina is a blue-green algae. We also call it as cyanobacteria. So this is rich in protein. It has around 51 to 71% of protein by dry weight. It's not only rich in protein, it also has the complete set of amino acids which are required for the human body, and it is rich in vitamins, minerals, antioxidants which are all healthy for the humans. So because of this, the spirulina is also used as a food supplement for the space travelers. You can find spirulina in the form of powder as well in the market. Question. The structure present in cyanobacteria helping in nitrogen fixation is haplosperm, holostrum, holotrema, heterocyst. The correct option is D, heterocyst. So the heterocysts are their special cells which you can find in few blue-green algae, basically filamentous blue-green algae. So the heterocyst, it can perform nitrogen fixation with the help of an enzyme called nitrogenase enzyme. Nitrogenase enzyme is active only in anaerobic condition. In the aerobic condition, the nitrogenase enzyme gets inactivated. In the presence of nitrogenase enzyme, the atmospheric nitrogen gets converted into ammonia, which is further used by the plants or the algae. 13th question. Plasmid is a double-stranded circular DNA, single-stranded circular DNA, single-stranded DNA, extra-chromosomal linear DNA. The correct answer is double-stranded circular DNA. If you take a bacteria, so we have the nucleoid. Along with the nucleoid, it has a extra double-stranded circular DNA which we call it as plasmid. So plasmid is an autonomous organelle. That means it can replicate on its own with the help of the host genome. So it can replicate on its own. You find the plasmid in the prokaryotes, and plasmid is extensively used in genetic engineering. It is used in the rDNA technology for gene cloning and etc. 14th question. Bacteria which reduces nitrates in soil is Nitrosomonas, Pseudomonas, Rhizobium, Clostridium. The correct option is Pseudomonas. These bacteria are also called as denitrifying bacteria because they convert nitrates into nitrogen gas. They work in anaerobic condition. That means in the absence of oxygen. And one more example for denitrifying bacteria is Theobacillus. So this is one more species which is a denitrifying bacteria. 15th question. Mycoplasma is not inhibited by penicillin as it lacks cell wall, ribosomes, nucleus, cell membrane. The correct option is A, cell wall. The mycoplasma is the smallest organism as we know, and it lacks cell wall. Many of the antibiotics, if you see, they target the cell wall synthesis. The penicillin also targets the cell wall synthesis only, but because it does not have cell wall, the penicillin cannot interfere with the mycoplasma's life cycle, and because of this very reason mycoplasma are resistant to many of the antibiotics. 16th question. Diatomaceous earth is obtained from Xanthophyceae, Basilarioophyceae, Chrysophyceae, Rhodophyceae. The correct option is C, Chrysophyceae. We know very well that this is formed from the cell walls of diatoms. So what are the cell walls of diatoms made up of? They are made up of silica, right? They are made up of silica. We also call the cell walls as frustules. So this is a different name. Now this cell walls, when the diatoms die, they settle on the bottom of the ocean. So over a billion years, what happens is this silica or this siliceous cell wall, it forms a sedimentary rock. Now this is what we call as diatomaceous earth, and this we use it for different purpose like filtering, polishing, and even for insulating materials. 17th question. Which of the following is not a character of Protista? Protists are eukaryotic, true. Some protists have cell walls, true. Body organization is cellular. One single cell can perform all the functions, right? Mode of nutrition is only autotrophic. This is not true because along with autotrophic, we also have heterotrophic. So what are the examples for heterotrophic mode of nutrition in Protista? That will be protozoa. Protozoans are heterotrophic. They are parasitic in nature. And we even have the mixotrophic. For mixotrophic, the example will be Euglena. Euglena, when it can perform both autotrophic and heterotrophic mode of nutrition. So mode of nutrition is only autotrophic is wrong. So the correct option will be D. This is not a character of Protista. 18th question. Which of the following groups of organisms have a protein rich layer called pellicle? Okay, so chrysophytes, euglenoids, dinoflagellates, slime molds. The correct answer is euglenoids. So instead of the cell wall, they have a protein rich layer called pellicle. This is flexible in nature. It's flexible in nature. 19th question. The red tide is caused by Noctulica, Goniolox, Gymnodinium, all of this. The correct option is B, Goniolox. Goniolegs are diatoms which cause these red tides by rapid increase in population. By rapid increase in population. So these are basically algal blooms which harm the aquatic animals. During this process, they even release neurotoxins like saxitoxin, which is very harmful for the aquatic animals. And if it gets into the food chain, it can even harm the humans. It can cause a paralytic shellfish poisoning in the humans. That's called as PSP, paralytic shellfish poisoning. So it's because of this Goniolux. If you see the ocean changes into red color. That is why we call them as red tides. 20th question. The reserve food in dinoflagellates is leucosin and volatin, paramylum, starch and oils, pyrenoids. This is very easy. The correct option is C, starch and oils. 21st question. Class Sporozoa of phylum Protozoa is characterized by flagella, cilia, pseudopodia, infectious spore-like stage. So the correct option is infectious spore-like stage. So we have an example also, that is Plasmodium, which comes under class Sporozoa, which causes malaria. 22nd question. Fungi usually store reserve food in the form of protein, lipid, starch, glycogen and oil. The correct option is glycogen and oil. So glycogen is a polysaccharide made up of glucose. 23rd question. Find the odd one with respect to fungi imperfecta. Alternaria, Penicillium, Colletotrichum, Trichoderma. So here Alternaria, Colletotrichum and Trichoderma, they are the examples of Deuteromycetes. So Deuteromycetes is another name for fungi imperfecta. So the correct answer will be Penicillium. Penicillium comes under Ascomycetes. It's not a Deuteromycetes. 24th question. Which of the following is used extensively in biochemical and genetic work? Aspergillus, claviceps, neurospora, puxunia. So the correct answer is neurospora. So neurospora is a model organism, acted as a model organism in research, mainly especially in the genetic work. 25th question. Which type of asexual spores are found in class Ascomycetes? Conidia, zoospores, aplanospores, both B and C. The correct option is it's A. Conidia are the asexual spores that you can find in the Ascomycetes. Like conidia, they are formed on the conidiophores. If this is the conidiophore, these are special myceliums. So upon this conidiophore, you have the conidia. So these are the conidia. 26th question. Which among the following can't produce spores sexually? Ascomycetes, Basidiomycetes, Phycomycetes, Deuteromycetes. The correct answer is Deuteromycetes. Because of this very reason, we also call them as fungi imperfecta, because we don't know the sexual phase of this Deuteromycetes. 27th question. Fusion of protoplasm between gametes is called plasmogamy, karyogamy, dikaryon, dikaryon phase? The correct answer is plasmogamy. Plasmogamy is the fusion of protoplast. And what is karyogamy? Karyogamy is the fusion of nucleus, right? And dikaryon stage is N plus N stage. That means when two different mycelium are combined, or then they have two nucleus. So that is N plus N condition. This is called as dikaryon phase. 28th question. With respect to fungal sexual cycle, choose the correct sequence of event involved. So here the events we know are, first is the plasmogamy. So that is the fusion of protoplast. And then we have the karyogamy, that is fusion of two nuclei. And finally we have the meiosis. So this gives the haploid spores finally. So just here we have plasmogamy, karyogamy, and option C is the answer. 29th question. An intervening dikaryotic stage occurs in, just now we have learned that dikaryotic stage means N plus N condition. The nuclei are not fusing, but they are separate within one single cell. That is called as dikaryotic stage. So this you can observe in the ascomycetes and basidiomycetes. So the correct option is B. 8th question. One of the major components of cell wall of most fungi is peptidoglycan, cellulose, hemicellulose, chitin. So the cell wall is made up of mainly chitin and polysaccharide. Right? So the major component is chitin. The correct option is D. 31st question. Which of the following statement is incorrect? Viroids are smaller than virus. That is true. Viroids are found to be free RNA. True. The RNA of the viroid is of high molecular weight. This is wrong. This is incorrect. So the viroid actually has a low molecular weight than the RNA. And then in 1971, T.O. Diener discovered it. That is true. So the correct option is C. The following disease is not caused by prions: BSE in cattle, CJD in humans, mad cow disease. All these three are caused by prions, whereas the potato spindle tuber disease, this is caused by viroids. So the correct answer will be option number D. So to talk about the prions, so prions are nothing but they are free proteins, the misfolded proteins. So they have the infectious, they act like a infectious agents. So these misfolded proteins act like infectious agent which can cause neurodegenerative diseases in both cattle and human. 33rd question. Select the wrong statement. So the viroids were discovered by Diener. It's correct. W.M. Stanley showed that viruses could be crystallides. Correct. The term contagium vivum fluidum was coined by M.W. Bajrik. It is correct. The mosaic disease is caused by bacteriophage. This is a wrong statement. It's not bacteriophage. It's the virus. So the correct answer is option D. The fourth question. The algal component of lichen is known as A and fungal component B. Identify A and B respectively. So algal component is what is called as phycobiont, and then the fungal component is called as mycobiont. Right? So the correct option will be option C, phycobiont. B is mycobiont. Last question. Identify ciliated protozoa: Radiolarian, Leishmania, Stentor, Monocystis. Write your answers in the comments.